day everyone. So today me and my teammates will be presenting on the topic of Escherichia coli. This is FATIN and this is CHUI. So first and foremost, I will leave it to CHUI to explain what is E. coli.
First is the E. coli. E. coli is a gram-negative gamma proteobacterium, which commonly found in the lower intestines of the warm blood organism. E. coli is rod-shaped organism. Now, E. coli is new star in the industry biotechnology field. This is because E. coli has the ability to transfer the DNA via the bacterial conjugation, transduction or transformation, which allow the genetic bacteria to switch the horizontal for an exciting conclusion. The optical floor of the E. coli is 37 degrees Celsius and can driven by aerobic and anaerobic respiration.
Again, I'm going to present E. coli timeline. E. coli was first discovered in 1885 by Julius, a bacteriologist, which is come from Germany. Then, next, in 1951, it was used to produce insulin and become most top study molecules in scientific history. Next, in 1960, the researching enzyme was discovered. As the time going down, a biochemist found a way to require a plasmid into an E. coli. Then, in 1970, E. coli being used in biotechnology for vaccine development, bioregulation, production of biofuel, production of immobilized enzyme, and in technologies of biocomputer also.
Why E. coli is important to the industry microbiology? First and foremost, the growth rate of the E. coli is fast, so that the production is higher. E. coli can be used to make the proteins. For example, enzyme and hormone. If we had this complex gene sequence, so that the modification process is easily. E. coli is easy to handle and this is also as a host. E. coli able to grow under both aerobic and anaerobic conditions. Besides that, E. coli is used because E. coli is safe and not harmful.
Now that we have understand why is E. coli so important to the modern industrial microbiology, I will be showing to you all how does E. coli actually contribute to the industrial microbiology. And the example that I will be showing will be the production of insulin. And before we go into that, I will be explaining the history of production of insulin. Insulin was firstly discovered in 1921, and sooner one of the diabetic teenager took the first injection in the next year. And then after 30 years, insulin became the first protein to be fully sequenced. And in 1960s, the insulin sources are still pigs and cattle because researchers at that time couldn't produce too much insulin in labs. And in the modern 1980s, the first human protein is successfully produced. And then after that, it will be using E. coli in this production of insulin. This will be the summary of the whole process for the production of insulin. First and foremost, we'll be isolating the gene from the gene donor, and also the plasmid from the plasmid donor, and in this case, it will be the E. coli. And after that, we'll be taking the isolated gene and combine it with the plasmid and form this recombinant DNA molecule. And after forming this recombinant molecule, we'll be replacing the original plasmid in the E. coli with this new recombinant DNA molecule. And whenever this recombinant DNA molecule will be reproducing, the donor gene will be inside this E. coli. And whenever this transcription and translation of the gene happens, the protein product will be also produced, which is in this case insulin. The 5 reasons that E. coli is being used in this production of insulin is that its competency as a host. It means that E. coli coli will not be harmed when we remove the original plasmid from its cell body. And next, it is due to its short doubling time, which is it will cause the high yield production. And also, its complete genome has been sequenced ever since 1997. That's why we are actually very easy to modify the whole gene, and we are free to put in whatever gene that we want. If let's say we wanted insulin gene to be inside, then we will just take out the insulin gene from the gene donor and we put it inside the plasmid. And next, it is due to this low cost of medium needed. The medium needed is actually glucose, because E. coli actually is okay with glucose. And lastly, it's due to its easy handling. It is suitable to grow in fermenters, and in that case, we are free to control the environment for it to grow. And also, it is easy to engineer to achieve genetic stability. Next, this photo shows the little girl before taking insulin and after taking insulin. And there are other ways E. coli contributed to industrial microbiology field that includes biofuel production, vaccine production, and enzyme production.
E. coli was very beneficial for us. It helped us to gain knowledge about microbiology. Then, actually, E. coli live in our intestine, so it helped our gut to digest food. Next, for microbiologists, E. coli help in the making of synthetic fuel, alcohol, and rubber. E. coli is a very easy to manipulate. Therefore, it is used for making of antibiotic, cancer-fighting drug, and insulin in medical field. Next, artists use E. coli and culture them in a petri dish until it's grown. Then it will become a beautiful picture. For